Yes, good afternoon everyone. So welcome back to Biochemistry Lab. So today I am going to perform another test which is for protein qualitative analysis. Okay, so next test which I am performing today is known as Xanthoproteic Test. So Xanthoproteic Test is basically for aromatic amino acids. If suppose you have a protein sample and you wanted to confirm what kind of amino acids are present within that sample, and if it is aromatic amino acids, then this test will come out to be positive. Okay, so already I have taken 1 ml of aromatic amino acids. So you know we have 3 aromatic amino acids: tyrosine, tryptophan and phenylalanine. So for all these 3 aromatic amino acids, this test come out positive. So we are going to learn how to perform aromatic test, or you can call it Xanthoproteic test. Okay, so theory part I have already told you. Because nitric acid is very concentrated, so you need to be careful while adding nitric acid. It can react and it can, if one drop fall into your clothes, so it can spoil your clothes. So it's very important whenever you are using nitric acid, be very careful about this. This is very dangerous. Okay, so I have already taken 1 ml of nitric acid. I have added into my sample. After that I will keep it in boiling water bath for 1 minute and I will observe the color. After that I will add NOH, sodium hydroxide, to this particular test tube. So let us go to water bath first of all. Okay, so we have already added this nitric acid to our 1 ml of sample. So I have taken protein sample. So if it will have aromatic amino acid, then this test will come out to be positive, and I am keeping my test tube for 1 minute inside boiling water bath. Okay, so we have to give it 1 minute time. After that I will take it out, then I will cool the test tube, then after that I will add sodium hydroxide and will observe yellow orange color PPT formation. So that will be okay. So 1 minute is over. So you can see now after 1 minute our test tube look like this. Okay, so this is our test tube. Now I am taking it out, and after taking it out we have to cool it for some time. Okay, so after cooling, after giving it some time for the cooling, then we have to add our next reagent, that is NaOH. Okay, so we will give some time for cooling. After that we will add NaOH to our this particular test tube, and then we will observe the PPT formation. Okay, so let's wait. Okay, so we have to we will wait for the final result, and final result will be orange red PPT formation. So that we will observe after 1 minute. So let us wait for 1 minute. When it will cool down, then we'll add sodium hydroxide, which I have already prepared, and I prepared it 40 gram of NaOH. Okay, so it is 40%. So I have dissolved 40 gram of NaOH in 100 ml of distilled water, but it is not total 100 ml, so I have prepared less volume. So less volume, 10 ml I prepared in 4 gram. So 4 gram of NaOH I have taken, NaOH sodium hydroxide pellets, and then I dissolved in 10 ml of distilled water, and that became 40%. So same percentage you have to keep depending on your final volume. You can vary, the concentration has to be same, but the gram weight you can vary according to your final volume. So let us add this. Now it is cool enough. Now we are going to add. So let me wash it first. Maybe there is. So washing is most important whenever you use any equipment, any pipette or anything. You have to wash that. Otherwise, you know, everything will be mixed. So I have washed it very carefully. Now I will add NaOH. Very slowly I will add to this test tube. So you can see there is formation of orange PPT. So you can see the change. Still it is not there, so we have to add more. it has to be alkaline enough so that there will be formation of orange red PPT. So you can see here color change, orange color. So we need to add little more. So little more I will add. So let us see what PPT we will receive. These experiments are very interesting. When you come to the lab and you perform yourself, you obviously learn. Okay, so now you can see the color change. See, this is orange red PPT formation has started. So let me add little more for your observation. Okay, so that will be our final observation. So that means this particular sample contain aromatic amino acid. Okay, so we have 3 aromatic amino acid, and to this sample I have taken tyrosine I have taken as a sample. So that is why this test will be positive. Okay, so I think it is enough. So earlier also you have observed, so here you can observe there is formation of orange red PPT. So this is our final observation for Xanthoproteic test. Okay, that's all about all the tests. So again you have to wash it very carefully, wash your pipette and everything whenever you use all the instrument. After that you have to carefully wash everything. You have to clean your workbench, and after cleaning your workbench and everything you have to maintain the hygiene, and you have to wash